Rendering, all right. Yes. Okay. Thank you. I want to emphasize that with Harper's work, that is like less than it's like 5 or 6 weeks worth of work by someone who did not know any Julia before she started. So it says a lot about what she accomplished. And it also tells you a little bit about what I'm going to talk about, which is Julia packages for genetic analysis and omic data. I want to emphasize that it is very easy to learn, and that is one of the things that was attractive to us. Um, so, this is basically an ad for all the packages that we have written in the past, 6-7 years. And they can be, the active packages that we have are in one of 3 categories: genetic mapping, matrix value data, and utilities. They're all related. I would say they're all related, and in general are dealing with large amounts like matrix value data in genetically variant populations. So the genetic mapping packages are bulk LMM for fast linear mixed model scans for large number of univariate traits with 1 degree of freedom. And there's a talk by Zifan Yu, Fred Yu later today on that. If you are interested in doing multivariate linear mixed models, there's FlexQTL. And if you want to do really fast linear model eQTL scans, then we have a package called LightQTL for that uses GPUs. This was there's 2 purposes: one, to see if we could do large amounts of these scans quickly, and this is really a precursor for bulk LMM. But it also was a platform for us to test out how to use GPUs, what's the benefit of using GPUs. And then we have 3 packages that are, and there's the Big River QTL plots, which is just to facilitate displays, and you saw a little bit of that. We have 3 packages on matrix value data. These are what I would call structured matrix value data. And they are organized around the idea of bilinear models. And if we have time, I'll try to explain what they are. And we have applied them to metabolomics, genetic screens, eQTL scans. A lot of familiar problems can be cast in terms of these bilinear models. And in order to do this work, you have to get data, manage it. And for that, we have built 2 API packages, one for Gene Network, and it is actually one of 3 API packages for Gene Network. The other 2 are in R and Python. This is in Julia. And we have another package for Metabolomics Workbench, and we have plans to add other services in the future. And finally, there's a package called Helium, which is just basically a binary format for tables. This is really a convenience packages for running simulations, storing data, fast things like that. And before I go further, I should say all of these, there's one person common to all of these, which is Gregory Farage. He's not here this minute, but I think you will see him later. And to various degrees, he has contributed to all of these packages. Okay, so why Julia? So the first motivation for us was, let's try something new. It looks interesting. It has a very pleasing syntax, at least in my opinion. I hope to show you. And the real selling point is that whatever you program, which looks like a prototype, will be quite fast, almost as fast as you had coded in C++. This is like a marketing, this is how they say, but this is more or less true. And the other thing is that it has very good support for parallel distributed computing. You have a lot of control over high performance internals that you normally don't get in these sort of high level languages. And finally, it's easy to learn. You can use abstractions using types and what's called multiple dispatch. I'll show you just a few toy examples to see how it works. And there's a very nice package manager. It's very easy to register packages. And there's a growing system of innovative packages. So as I was saying, with parallel computing, it's very easy to distribute jobs. Linear algebra is multi-threaded by default. So, things like matrix multiplication work really, really fast. But you can also take control of that process. So that's, anyway. So here's the kind of 3 use cases. So one use case is tool builders, such as myself. We want to design an algorithm, make something that works, and we don't know what we are doing. And what's in it for us is what will be called a very expressive language. And you can build abstractions. You can encode abstract concepts and build on it. For example, I'll show you a little later with distributions, statisticians, like we can make certain operations on distributions. You can add 2 random variables. You can multiply a random variable. Those concepts can be encoded very easily in this language. It has excellent development and runtime speeds. One example is, someone like Harper was able to make progress very quickly, very fast development times. But things run pretty fast, as you saw, on even a very modest hardware such as her laptop. Now, the other extreme is non-programmers. What if you're not interested in programming? And for that, reactive interfaces like Pluto notebooks, like what you just saw, you can use that. If you are just a little bit more curious than that, you can modify that notebook. What she showed earlier can also be modified. If you don't like the interface, you want a different option, you can tweak it with a very good starting point. And finally, I think are the data analysts, and I think this is maybe a large fraction of users. I would say, if you're happy with whatever system, don't change. You can be where you are. But if you are unhappy, need greater speeds, or you're managing really large data sets, then Julia might be worth a look. But you can also call Julia from R, and also it works the other way around. If you're using Julia, you have a favorite R package, you can also call that R package as well. So here's a quick look at the language just to show you what it looks like. So here is a very simple function, like I define a function fx equals to x squared plus x plus 1, just like you would write it on paper. Same thing, right? So easy. And I say plot f from -4 to 4 and give it a label x squared. Notice it's not x2, but actually x squared written properly. So it's just very easy, okay? So this is just an example of how you would define and use a function. And here's an example of an abstract. So distributions are, statistical or probabilistic concept. In a language like R, if you want to take the PDF of a normal distribution, you have one function. If you have chi-square, you have another function. But conceptually, they are related, right? You want a certain thing from different distributions. So in Julia, it is very streamlined. I have a function called CDF. I pass it a normal distribution. It returns 0.5 at 0.0, standard normal. If I want to do the CDF of an exponential distribution, I just pass that to the same function. So it's, I can also do things like mean of that normal distribution and give you the correct answer. Mean of a normal with, mean 0 and variance 1 is 2. So that's what it gives you. But here is the more interesting thing. You can do more complicated things. So what I'm doing in this 2 lines of code is I am taking the operator plus, and I'm telling it that if you have 2 distributions, the plus means convolve the distribution. That is, add 2 independent versions of that distribution. So this gives very interesting results, just like 1 line of code there, or 2 lines of code there. I say, add a chi-square with 1 degree of freedom to a chi-square with 2 degrees of freedom, and it gives the correct answer, chi-square with 3 degrees of freedom. Okay, this is just beautiful. But this is really a toy example. Probably you can do it in any language with a little bit of work, but it shows the power of using abstractions and building on it. And it also knows that if I say twice a normal distribution is not the same as adding 2 normals. So if I say twice a normal, it'll give you a normal with mean 0 and sigma 2, standard deviation 2. But if I add 2 independent normals, which is the plus, so it's not, then it gives the right answer, that is mean 0 and standard deviation of square root of 2. So I think this is just in my, it's just beautiful that it works this way. Okay, linear algebra, the bread and butter of what we do. If I have a, I just simulate a very large matrix like this, random. This is 1 function. I give it, I could change this to exponential if I wanted. And I say x transpose x looks exactly like on paper what you would write. And in half a second, it gives the answer. And if you want to do x trans, this is like x, something more complicated. It's pretty much whatever you write on paper is what you're writing in code, and it works. I think that's what I want to say here. Okay, so here next I'll go through the packages that we have in a little more detail. So I told you about bulk LMM, and there's a whole talk devoted to it, work of Fred and Gregory. The design goal is moderate sample sizes, such as what we have in model organisms, but many, many traits. It makes the assumption of 1 degree of freedom. So it will work for collaborative cross, not on the haplotype, but only on the SNPs, and no missing data. So, we have to make some compromises in order to get the speed. There is support for permutation tests, and you can use it for simulation studies. You can weight observations, and you can also do a little bit of Bayesian, like add a prior on the residual variance. You'll hear more about that later today. I told you about the multivariate linear mixed model genome scans. That's the work of Hyunju Kim. And this is a generalization of what you have in the multivariate version of GEMMA. It uses a accelerated gradient descent algorithm and can handle, over 20 traits simultaneously. I will skip that. So next, I want to tell you about matrix valued data and bilinear models. So the concept here is that you have a something from a high throughput technology like metabolomics, and there's a matrix called Y, and you have sample characteristics, whether or not a certain individual has a certain genotype or not. That's a matrix X on the side, But you can also think of these metabolites, and I'll take the example of triglycerides. So when we are analyzing data, we think of the individuals as coming from a population, and I want to see what's the difference between individuals with this genotype versus that. You can also think of the metabolites as a population. You have a population of triglycerides, and you want to see how triglycerides with a certain characteristic behave differently from another, right? That is what you can do using the framework of these bilinear models. So it's kind of linear in two directions. So that's the gist of it. And so here is a triglyceride. You have a glycerol backbone, three fatty acids, and then you can have 0 to whatever number of double bonds, indicating the degree of saturation. So I'll show you what the kind of thing it can do. So this is from a human study. The individual characteristic is whether or not the individual is taking fish oil or not, okay? And so what I have plotted on the left is the correlation between the triglycerides, which, as you can see, is very complex. They are not independent. And on the right-hand side, I have plotted each point is a triglyceride, and the color indicates to what extent the level was higher in those taking fish oil versus not. So immediately you can see there is a correlation between the number of double bonds you find in a triglyceride and the total number of carbons. They are correlated. If you notice the structure, it makes sense. The longer the fatty acid, the more carbons it has, the more chances of a double bond. So, but it is clear from this picture that the strength of the association is driven by the number of double bonds or the degree of saturation, and not the chain length, the number of carbons. But if you look vertically, you might think that longer triglyceride with longer chains have higher levels. But it's really, if you look more carefully, it is really driven by the number of double bonds. And that is the kind of thing that you can disentangle using a bilinear model. And here's a view of that. The unadjusted analysis would show that there's an association with the number of double bonds as well as the number of carbons, but an adjusted analysis where you simultaneously consider the number of double bonds as well as the number of carbons tells you that no, the effect is not driven by the number of carbons, by just the number of double bonds. So, and that's what we have a couple packages about that, and I told you about the other API packages. And looking forward, we want to build upon. We want maybe make a QTL mapping meta package. Big River is the brand for that, because in Julia you have to name packages so that there is no ambiguity, and the experimental packages will be called Wolf River. Wolf River flows into Big River. And we want to build on these teaching resources and interactive notebooks. So in summary, Julia is an attractive language for biomedical data science. Take a look. We are happy to talk about it. Short development times, fast speed. And this is my thanks web and a list of our resources. And we are looking for people to join us. So if you know someone, please, I'll be happy to talk. Thank you.